let's talk about bottlenecks. When it comes to lentiviral vector manufacturing, excuse me, such as transfection efficiency, yield, cost. What are some of the best practices? First of all, what are the major challenges, and what are the best practices to perhaps even attempt to overcome them?
Yeah. So typical way of manufacture is to actually transfect mammalian cells, and that's typically a 4 plasmid transfection system. So while it's entirely doable, there's of course a lot of cost and time to make GMP grade plasmids and to have to do multiple ones. It's so to improve that, there are packaging cell lines that are needed because some of the factors involved in production are cytotoxic. You need to then make them inducible expression. So you have more engineering to go. And then you can introduce the gene of interest by transfection of a single plasmid, or you can eventually introduce that as, again, part of a producer cell line. But it does take about 6 months to produce a producer cell line and another 6 months to get to a GMP cell bank. So it takes time to start using those methods too. Obviously, if it's something you're continuing to make, that is a really great way to go for future production. And the other piece of it is also ensuring that you move to suspension cells early in development, because again, it's much harder to scale up with adherent cells. So in general, moving to suspension cells earlier in process development can also help improve your efficiency and scalability.
Good. Okay. Now, how would you say the field is evolving in terms of pseudo-typing? So, to improve tropism, reduce immune responses for lentiviral vectors. And this is a question too that came in from our registrants. So, more than a few people were wondering about this one.
Oh, interesting. Yeah. So there are new virus envelopes that are being used. So there are some interesting ones coming through that do bind to different receptors, and so may somewhat change the tropism or allow you to do a different type of targeting. And even the COVID spike glycoprotein's been used there. The other interesting thing that is being worked on is to also either create mutations in existing envelope proteins that are being used, or inserting ligands. Similar to what's being done to retarget AAVs, you can potentially insert ligands into these lentivirals envelope proteins and use those to target certain cell types. So that is coming forward. And again, with some of these, you're going to have fewer pre-existing immune responses. So that could help for in vivo gene therapy also.
Great. Okay. Given your knowledge and expertise in AAV, what are the main safety and durability advantages lentiviral vectors offer over AAV and perhaps adenoviral vectors? Are there limits to their scalability or manufacturing that developers perhaps should be aware of?
Yeah, so in terms of durability, what lentivirus gives you that other vectors don't is durability even in dividing cells. So it gets, moves on to all the daughter cells. So where that can be useful, of course, is in bone marrow derived cells. And even you can consider other tissues that might divide actively, such as a baby's liver. As it's growing, you could carry on gene expression. So if it gets into a cell, it typically integrates and it will be stable there and be able to be passed on to daughter cells. So that is one area in which you really want to consider that. Again, depending on what disease you're trying to treat, what's your therapeutic approach? Do you want it there permanently, as we would do with a gene replacement therapy? If you're doing something else like genome editing, you typically want the genome editing machinery to come in, do its job, and then kind of go away. You don't want it sitting around trying to keep editing and editing. And so there you might choose a different type of vector in which you will get transient expression. Then that's desirable. In general, in terms of safety, So it's an interesting conundrum. So with some of the vectors, such as AAV, when you inject them intravenously, you can, and with adenovirus too, you do get immune responses to them, which can be helpful or harmful depending on the type of immune response and the scale of it. So for lentiviral vectors, when they're used ex vivo, that becomes of very little concern, because it's already into the cell by the time the cell comes back gets reintroduced into the body. And so you're not exposing the body to large amounts of foreign virus. It has its own disadvantages, of course, in how difficult and complex that is, because for every patient, you're creating a bespoke product, which is their own stem cells that have been transduced and that are reintroduced. So you have to take all of those factors into account. And that the ex vivo gene therapy approach too, in terms of manufacturing, is more complex, because you're creating a new manufacturing run for each patient in order to get those cells ready. So for in vivo gene therapy, if it evolves towards in vivo gene therapy, some of those limitations will come down.
Okay, thank you so much. Please do feel free to submit your questions about lentiviral vectors. We're going to move on now to adenoviral, but happy to, like I said, answer questions from our audience about specifically for Karen about lentiviral. Moving on to adenoviral. And Francesca, I'd like to start with you, please. What do you see as the most promising use cases for adenoviral vectors today? Oncolytic viral therapy, gene therapy, vaccines, what have you, and why?
So for us, our major experience is with this vector. It's called CAN2409. It's a virus that has a little bit of a combination in terms of, we call it, in fact, a multimodal immunotherapy. Is directly delivered into the tumor. It's a replication defective adenovirus, so the safety of this, it doesn't integrate into the genome, but it transcribes a TK, so the thymidine kinase derived from HSV. And when it's administered together with a prodrug that is called valacyclovir, that is a generic, that is given for 15 days orally after the administration of the virus. So what happens there is that the thymidine kinase activate the prodrug and forms a metabolite that integrates into the DNA and induce immunogenic cell death. So it's multimodal in the sense that is oncolytic in its activity, but at the same time, the virus itself has got a series of proteins in the capsid that are extremely immunogenic. So what happens is the mechanism of action activates, releases in the tumor microenvironment, the tumor own antigen. So it's like an in situ vaccination. And at the same time, the capsid, the protein of the adenovirus, really activate the tumor microenvironment. So really is both an oncolytic and a vaccine at the same time. So I think that for us is an interesting modality. It's completely new in the way in which it's been applied to clinical trials. And we've seen that it's also extremely synergistic with standard of care, what we were discussing before about the fact that it's intratumorally delivered, meaning that it's got a very good safety profile and can be easily overlaid to standard of care, synergistic with standard of care, so we can use it in combination with surgery, chemotherapy, radiotherapy. And in the radiotherapy, we just reported the data from a phase 3 trial, in which we demonstrated that in localized prostate cancer, a patient that received CAN2409 plus prodrug on the top of radiotherapy were protected from tumor recurrence up to 30% more than patients that only had standard of care radiotherapy. So extremely interesting mechanism of action. For the HSV, the mechanism of action, we have one product in development that is really a classical oncolytic. That is a virus that actually is a first-in-class in its modality, because it's the only oncolytic virus that retains the ICP34.5. That is a gene that is classically associated with neurovirulence, but is also a gene that really supports tumor replication. And what we've done is to protect the neurovirulence effect, is that we have encoded this gene. We have deleted the 2 native forms of this gene, and we've reinserted them under a specific promoter that is the nestin promoter. Nestin is highly expressed in recurrent high-grade glioma, in glioma in general, so a brain tumor. but is also highly expressed in other very aggressive form of tumor. So we can control the replication of this virus in these settings. And then we have other platforms that are HSV related. We can talk about it later.
Sure. Yeah. Looking forward to that. We have an entire section dedicated to it. So that's good. Karen, before we move on to our next question, I would like to get your perspective on this. What do you see as the most promising use cases for adenoviral vectors today?
Yeah, so I absolutely what Francesca was talking about are really great uses of it. So both for oncolytics and potentially for vaccines, although it's, there's been a little bit of mixed data over the years with it. I think the other place to mention is that because expression from the adenovirus is typically transient, it can be useful for indications where you do want transient expression. So, for example, if you want to transiently express VEGF to restore blood flow to an area of the body, that is one potential way to get it in. And they certainly are, as a vector, it's more stable than injecting an mRNA, for example. So I think it can also be considered for gene therapies where you want transient expression.